Swayam Prabha. Digital India. Educated India.
Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and let's continue our discussion about the polymerase chain reactions. So, in our previous lecture what we have discussed about the different steps and how the technique has been evolved in a span of couple of decades, and how the polymerase chain reaction is being inspired from the DNA replications, and how the people have mimic the similar kind of steps under the in vitro conditions to amplify a shorter stretch of DNA using the site specific primers. So, in the today's lecture what we are going to discuss about the technical aspects of the polymerase chain reactions. And today's lecture what we are going to discuss about the reagents which are important for the polymerase chain reaction, such as the template DNA. Then we are going to discuss about the primer designing more in an interactive way, so that you will be able to design the primer using the available softwares. Then we will also going to show you with the help of one of the softwares how to do the analysis part, and then at the end we are going to show you the how to set up the PCR reactions, how to add the individual components, what are the precautions you should take while you are setting up the PCR reactions, and we will take you to our lab and we will show you how to set up these reactions. In addition, how to set up the different steps within the thermal cyclers, and at the end we will also going to show you the demo how to prepare the and set up the PCR reactions. And once the PCR reactions are over, then we will take out those reaction products and analyze them onto the agarose gels, and at the end of this lecture we are also going to discuss about the potentials of PCR in biomedical research or biotechnology research. So, let us continue with the template DNA. The template DNA, as we discussed in the previous lecture, could be genomic DNA fragments which you can isolate from the environment as well as from the biological samples. You can use the plasmids or you can use the recombinant DNA, you can use the viruses, you can use the tissue samples, and the only thing what makes a sample good or the bad is that the sample what you use in the PCR reaction should not have the nicks, or it should not be a degraded form, because if you are using the site specific primers and if there is a degradation into your gene of interest, then the PCR amplification may not happen. And apart from that, when you have a degraded samples, the degraded sample sometime always gives the non-specific reactions as well. This also we have discussed the primer designing, we have discussed about designing the forward primers as well as designing the reverse primers. So, what we have done is we have taken you to our laboratory, and where the my student Suram Banesh has prepared a small movie, and with the help of the softwares what he is going to show you is that how to prepare or design the oligoprimers or oligonucleotides, how to analyze them using the Sipson softwares under the different parameters. If you recall, in our previous lecture we have discussed about the primer length, primer TM, the annealing temperatures, GC content, GC clamps, and all these aspects he is going to show you with the help of that particular softwares. Apart from that, he also going to show you the different types of defects which you can encounter into designing of these primers, such as the primer dimers or the loop formations, and how to get rid of these loops. suppose you have a loop and that's how the Banesh is going to give you a demo about the primer designing and the analysis of these primer sequences.
In this video, we will show you how to design primers and analyze for different constants like hairpin loops and primer dimers, how to analyze these things using various online tools available. Hello everyone, in this video I will show you how to design the primers and analyze them. So for designing primers, first you have to identify the region of interest, your region of interest which you want to amplify from any vector or any sequence. So in second step you have to identify non-cutters. There are various softwares available, but we can use New England Biolabs NEB cutter version 2.0. After identifying non-cutters, you have to select a suitable vector in which you want to integrate this amplified region and suitable restriction sites. You will get suitable restriction sites from non-cutters. After that, you can go for designing forward primer. So for understanding purpose, I gave this sequence. So I am using this sequence. I will use this sequence to design the primers and analyze the primers. So this is the whole sequence, but I don't want to amplify whole region. I want to amplify the letters, the sequence which is highlighted in green. So I want to amplify starting from here to here. So now the question arises, what are the non-cutters? So you want to amplify this region and integrate into another vector. For that you have to identify which are non-cutting restriction enzymes. So what I will do, I will copy this sequence into NEB cutter and identify what are the non-cutters. So I just copy the sequence and paste here and I will ask submit. So it will analyze the sequence and give non-cutter. These are the enzymes cutting inside the sequence, but we are interested in which are non-cutters. So that means you can see here non-cutters. So just click here. It will give a number of enzymes which will not cut inside the sequence. So once getting this list, we have to identify in which vector you want to integrate your amplified region. So for that purpose, so I have selected, for easy of understanding, I have selected PET23A vector. So you can see this is the vector map. So this is the 5' side, this is the 3' side, N terminal, and this is the C terminal side. N terminal means forward primer, C terminal means reverse primer. So I can use BOM-H1 in forward primer and XH1 in reverse primer. This is the detailed map. So I have identified two restriction enzymes, that is BOM-H1 and XH1. So I can use these enzymes in forward primer and reverse primer. So after identifying restriction enzymes and the vector, we will go for designing forward primer. So I will take this sequence. I want to amplify from here to here. So I will copy this sequence here. So for designing forward primer, it is very easy. You have to take the sequence, whatever you are getting, up to 15 to 20 bases you can take as it is. So if you want to insert a restriction enzyme, suppose I want to insert a restriction enzyme, this is the sequence as it is given from this this whole sequence. So I want to insert a restriction enzyme, that is BAM-H1. So this is the sequence for BAM-H1. Here it cuts, so I can use this sequence here. So this is our restriction enzyme. Here it will cut. So we cannot simply give like this. So there should be some more bases, extra bases we have to add in the 5' side. So I will use so this sequence I will use. So now this is 5' to 3' side. So this is our forward primer is ready. So after designing this forward primer, we have to analyze this sequence, so this primer. So what I will do is I just copy this sequence and I will use Oligo Analyzer software, which is specially designed for this purpose only. I will paste the sequence, just ask analyze. So here also you can see there are so many options are there, like you can analyze hairpin loop, self dimer, heterodimer. So these are the general details: what is the length and GC content, melting temperature, molecular weight. So these are normal details. I will go for hairpin loop. Is there any hairpin loops? So we can see there are number of hairpin loops. We can see different structures predicted by the software. So if you want to explore this thing, you can explore only two bases it is forming, and the delta G value is minus 0.43 kilocalorie per mole. So this is fine. Up to minus 10 kilocalorie per mole is fine. Those hairpin loops broken during the amplification process, but above minus 10 kilocalorie per mole cannot be broken. So in that case, what we will do, either we redesign the primers, or we will add 5% 1% B10 or 5% DMSO. These chemicals disrupt these loops, so that the amplification will be fine. So next I will analyze for self dimer. Is there any self dimers, and what is the maximum delta G? So this is forming continuously 5 bases. It is because of the restriction sites. So those are restriction site. Those homodimers forming due to restriction site can be broken. There is no issue. But other than that, this is also because of restriction site. But other than that, we have to look carefully. So is there any continuously 4 or 5 bases forming this homodimer? Then it is very difficult. These interactions can be broken easily. So here some of the consecutive base pairs are there. These are very weak interactions, so they can be broken. So other than that, there is no significant self dimers. So this sequence can be used. And for heterodimer, predicting heterodimer, you need a complementary sequence with reverse primer, like reverse primer you need. So that we will discuss later on. So we got our forward primer here. So it is very easy to generate forward primer. But in case of reverse primer, it is somewhat difficult, because not in terms of predicting things, it is somewhat tricky. So what I'm saying is, here we have sequence. So in case of forward primer, we just take an as it is sequence, 15 to 20 bases as it is from sequence itself. But here we have to take complementary sequence, not 3' to 5' or 5' to 3' sequence. We have to take complementary to this one. Say this is the sequence we got from here. So what is the complementary to this one? So just I will add here. So this is the complementary to this particular sequence. So as you can see, this is we have to keep from this direction, 5' to 3'. So I will take like this. So what we have to do is, we want to insert a restriction site here. So we can insert a restriction site here directly. So in reverse primer, we wanted to insert XH1 site. So this is the restriction site. As usual, we can use we have to insert T here. So this is the restriction site we added. We can add flanking regions in between, flanking bases before this restriction site. So now we got our reverse primer. So we have to go through same procedure like what I have shown in case of forward primer. So just I will copy paste here and analyze the reverse primer. So is there any hairpin loops? Only one hairpin loop, that is within the range of delta G, so there is no issue. And self dimer. So we can see here continuously 4 bases are forming. In this case we have to either change the sequence or remove some of the bases. We can ignore those dimers forming through restriction site. So next, heterodimer we have to analyze. For heterodimer we need forward primer. Just copy, paste here and calculate. It will give is there any heterodimers. This is because of restriction site. This is also because of restriction site. This can be broken. Those which are at the end of the sequence, they can be broken, but which is in middle, if those bases are middle, it is very hard to disrupt those interactions, and our amplification will be not good. So there is no amplification literally. Other kind of interactions will be broken easily. These are weak interactions. So this is how we can prepare, design the primers and analyze the primers. We have done all these processes for designing forward and reverse primers, but instead of doing manually, we can do it online. We just have to submit the sequence and it will return the forward and reverse primers. These are some of the tools available online for freely, but there are commercial tools also available like Algo 7 Vector NTI Primer Firmware. So if you interested in these softwares, or you can just go through these sites and submit your sequence, you will get your primers.
And then subsequently we are going to show you how to set up the PCR reactions. This is a typical thermal cyclers. We have already discussed the different components of thermal cyclers, and what we have done is we have also prepared a very small movie from the thermal cycler what is available in our laboratory, and in that particular small clip what students are going to show you is about the different components which are present in a typical thermal cycler, such as the reaction vessels or the place where you are going to keep the reactions, or the heating block which does not allow the evaporation of the sample, and the different types of buttons what are available for different types of operations within the machine. Apart from that, the students will also show you how to set up the PCR reactions. So the purpose of these videos or the demo is to show you the how to perform these experiments.
In this video, we will be demonstrating how to set up a PCR reaction and analyze the results using agarose gel electrophoresis. PCR or polymerase chain reaction is a widely used molecular biology technique to amplify a particular segment of DNA. It is also employed in biomedical research and forensic medicine. The main application of this polymerase chain reaction is cloning. To set up a PCR reaction, we need template DNA, site-specific primers, DNTP mix, nucleus-free water, and Taq polymerase. For a 50 microliter reaction, in a typical concentrations of 10 to 100 nanograms of template DNA used, and 5 picomoles of each primer will be used. This is a earlier version of thermal cycler which contains display unit where we can observe the parameters and change the parameters. This is a hot shield, this is sample holder, and inside there is a Peltier system which can maintain the temperature fluctuations. For setting up a PCR reaction, initial denaturation at 95 degrees Celsius 3 minutes, and these steps we will use 30 repeats where initial denaturation will be 30 seconds and annealing it extension time should be given 1 minute per kb. And here final extension should be given 10 minutes and hold it 4 degrees Celsius .
Now moving on to the third thing is about the PCR reactions. So, not only they will let you to discuss the PCR reaction, they will also let you to discuss about how to set up the cycles onto the machine and how to operate them. And this operation could be the machine what we have in our laboratory may not be the machine what is available in your institutions or in your setup, but the more or less the steps are remain constant, whether it is a machine from the one company or the other company. Only the few the buttons may be different, or few the way you have to operate these machines may be different, but the overall basic principle remains a constant. Then at the end the Banesh and his team is going to show you how to analyze the PCR product. So, what you are supposed to do is, once the PCR is over, then you are going to take out the sample from the PCR machines. Then you have to cast a agarose gel, and agarose gel, while you are casting the agarose gel, you have to take a lot of precautions, and then while you are casting, you can add the dye which is called as the ethidium bromide. So, ethidium bromide is a intercalating agents, and it intercalates into the DNA, and that is how it gives fluorescence into the DNA. So, once the gel is ready, you can load your sample into the wells, and you can run the samples and visualize it under the UV transilluminators, and you can take the images. Once you take the images, the students will also show you how to analyze these DNA, both for the size as well as for the amount of DNA present in your gel.
Once the PCR reaction is completed, we have to analyze the results for amplification. For that we need agarose and TAE buffer. First we have to weigh agarose and mix with the TAE buffer. It will not dissolve easily, so we have to heat it in microwave oven until it gets dissolved. Now agarose got dissolved in TAE buffer. We have to let it cool down up to 50 degrees Celsius. Now before pouring we have to add ethidium bromide for detection purpose. Now the gel got solidified. We have to take out the gel and keep it in the electrophoretic apparatus. We have to gently remove the comb loose the knobs and keep the gel in the apparatus. Make sure that the buffer is submerged the gel. We have to fill the remaining part with 1x TAE buffer. Generally for analyzing the DNA samples we will use agarose gel electrophoresis. This is the power pack and this is the electrophoretic apparatus. This is a negative electrode and this is a positive electrode. We can change the voltage from here. For loading up sample, we have to mix PCR reaction mixture with 5x loading line. The loading is over. We have to cover the electrophoretic apparatus with the lid and we have to adjust the voltage. Then start run. After the agarose gel electrophoresis, we have to visualize the amplified product. This is the Chemidoc MP where we are going to visualize the amplified product. Now we have to keep the gel then close the thing. We have to select here application nucleic acids with ethidium bromide, exposure, optimal exposure, or we can select manual also. Then we will acquire the images. Now we can find here, this is the DNA ladder, this is the PCR amplified product. We can transform or save this image into JPG.
Now at the end we would like to discuss about the application of PCR in different fields. So the PCR has the vast applications, whether it is a food science, whether it is a plant tissue culture, or it is a parental testing, means the all sort of genetic investigations, or whether it is criminal investigations, or whether you are going to use the PCR for the diagnostic purpose, or whether you will use the PCR into identifying a plant species or characterizing a plant species, that this is actually the particular plant species, because many times the molecular markers what you are going to use to identify a particular gene or particular plant is more specific compared to the taxonomical evidences. So let us discuss few of them. So PCR is extensively being used for diagnostic purpose. In this particular example, we have taken an infectious disease which is called as the HIV, and what you are supposed to do is, suppose a patient is infected with HIV, or it is a sample where you have to detect. So what you are supposed to do is, first you extract the blood from the patient, and from the blood what you are going to do is you are going to perform the PCR analysis for the HIV virus. So you have the site specific primers which you can use to amplify the viral DNA, and if the sample is going to show you a PCR product for the virus DNA, then the sample is considered to be positive. If it is going to show you negative result, then it is going to be negative for the particular disease. It could be the HIV, it could be viral diseases such as the hepatitis B and C, it could be human papilloma virus, it could be Mycobacterium tuberculosis, and so on, that you cannot use the PCR based diagnostics for any organism. You can use it for bacterial species, you can use for viral species, you can use it for fungal species, as long as you will be able to characterize the particular type of molecular marker or particular type of DNA sequence which is specific to that particular organism, and that may not be present in other organisms. So as long as you have that specific sequence, you could be able to identify that particular sample by doing the PCR amplifications. So the sample, the way you have to do is analysis is very much similar, that you are going to get the organism and then you will do the PCR amplifications. Now, in many of the cases, the parental DNA or the parental testing is also one of the major reason or major area where the PCR is contributing into the biomedical applications. So with the help of the PCR, you can be able to assess whether a child is belonging to the given parents or not. You can see that for that particular kind of analysis, what you are supposed to do is, first you have to characterize the DNA marker what is present in that particular family, and once you characterize those DNA markers, what you have to do is you have to see whether those DNA markers are present in the child and as well as into the mother and father. So what you see here is that the marker A is present in mother 2631, whereas in the case of father it is 29 and 30. So you can imagine, if it is a child of these two mother, then the child should have the combination of these DNA markers of mother or the father. So, you can see that the child is having a marker which is of 26 and 30, which means it is getting the 26 from the mother and the 30 from the father. So, that means it is potentially being a child of these two, mother and father. Similarly, you can see that the mother has the marker for 8 and 9, whereas the father has for 10 and 11, and the child has 9 and 10, which means the 9 it is getting from the mother and 10 it is getting from the father, which means it is actually be a potential child of these two parents. Similarly, you can see the all other combinations where the child is showing the markers which are of the mixture of these two parents. So, the PCR is a very powerful technology to detect the diseases for genetic testing as well as for the parental testing. For example, if somebody is prone to the cancer because it has a BRCA gene, that the BRCA gene PCR amplification also can be done to predict whether a particular person is predisposed to the breast cancer or not using this PCR technique. Then the PCR is a very powerful technique for criminal investigations. In the criminal investigations, what you have is you have the blood, hair or the skin sample from the side of crime, and from any of these sources you will get the cells, means you will get the DNA, and once you get the DNA, what you can do is you can do a PCR amplifications and generate the particular type of pattern. So, what you can do is you can see that from the crime scene you got the DNA which is giving a pattern like this. Now, what you are going to do is you are going to analyze the blood of the suspects, which means in this case we are showing the 4 suspects, suspects number 1, 2, 3 and 4, and what you can see is that the suspect number 1 is showing a pattern which is not completely matching with the crimes, the DNA what you got from the crime scene. Similarly, for the suspect 2, the pattern is not matching with any of these DNA what you got from the crime scene, and the suspect 4 is also not matching. But what you see is that the suspect 3 is showing a DNA pattern which is exactly matching with the DNA what you got from the crime scene, which means this particular guy is the potential convict and the potential criminal who probably have done this particular crime. And the PCR mediated evidences are very strong, very reliable, and they are being tested on several time or several attempts. So, that is why the judiciary system as well as the criminal system is utilizing and relying on the PCR mediated investigations, and the result what you get is very reliable. With this we have concluded our discussion about the isolation of gene of interest. What we have discussed so far, we have discussed that the gene sequence can be isolated under the 2 different aspects, whether the gene sequence is known or whether the gene sequence is not known. Okay. And in both of these cases, when the gene sequence is not known, you can have the flexibility of using the genomic library or to the cDNA library, and subsequently you will get the desired gene fragments, and that can be subcloned and can be used for downstream applications. Similarly, if the gene sequence is known, you can use the PCR as a technique to get the DNA fragments, and then these DNA fragments can be cloned into the expression vectors for the downstream applications. So, with this I would like to conclude our lecture here, and in our subsequent lecture we are going to discuss about the how this particular fragment what you got from the after isolating the fragment from the either genomic library, cDNA library, or utilizing the PCR as a technique, how to clone this particular fragment into your desirable transforming agent such as the plasmids. So, with this I would like to conclude our lecture here. Thank you.